It's electric.
So in week 4, we're starting to study neural development. And I just want to say that the book the book takes 3 chapters and over 100 pages dedicated to neural development. So it's a huge area of study. And I'm trying to pick and choose which of the subtopics within this area to sort of for us to take a closer examination at. And so I will likely focus on not the entire chapter for each one of these, but really sort of specific, and we'll go sort of like deep in some areas and then coast on the surface of some others. So let's just show a picture. This is from the textbook. This is the first figure in the chapter 13. And essentially what's shown here is neural development sort of from a view of 20, 30,000 feet. What they're showing you is that we want to look at how these cells, right, are going to become the neural tube, how those cells then differentiate into cells that migrate away from what will be the spinal cord, how they differentiate into other sorts of neurons, how they differentiate into cells that are bringing information to and from the periphery, how the cells develop into this central nervous system, and then what neural crest cells do. So with that as a frame, let's take a look at the second figure. Back when I talked about gastrulation, I gave you the 3 germ layers as being the ectoderm, the mesoderm, and the endoderm. And honestly, we're going to focus for much of our study of development on the ectoderm. And the ectoderm, let me get that here. The ectoderm, I told you I gave you a couple of cursory sorts of things that it becomes, but this is a little bit more specific. It'll develop into sort of 3 main tissue types. One is what we call the epidermis, or the surface ectoderm. And then if you look here to the right, here's all the different sort of subgroups of tissue types that is. Hair, nails, glands, the epithelial lining of your mouth, the lens of your eye, the cornea, tooth enamel, blah, blah, blah. There's another set of cells that derive from the ectoderm, and they're called neural crest cells. They're going to become the peripheral nervous system. And the peripheral nervous system is basically the part of the nervous system that is taking information from the peripheral, so sensory information, bringing it into the central nervous system, and then also that part of the nervous system that goes out to respond to that sensory information, so it might be muscle cells. Also the adrenal medulla, melanocytes, different other parts of the teeth, things are called glial cells and Schwann cells. Those are specialized neuronal cells that the Schwann cells are basically the insulation for nerve cells, and we'll talk more about that later. And then we also get the sympathetic and the parasympathetic nervous system. And then finally, the third is the neural tube and the neural plate. I'll show you that video again I showed in class, but that is basically the central nervous system except for motor neurons. So the brain, spinal cord, retina, and then motor neurons, which go from the spinal cord out into the periphery. So let's take a close look at that then. Before we do, I just want to say one thing about sort of neural development. Here's sort of just a schematic of the central and peripheral nervous system. And so the brain, spinal cord, right up here. I think I want to show you, like, it's pretty amazing. The spinal cord only goes to about this level, right? And yet neurons are going to have to exit this spinal cord. So think about in development, for example. And we've got sensory and motor neurons that are going to be, let's say, as far away as where your big toe is. And there are single individual cells, single neurons that are going from, let's say, a sensory receptor in your big toe. It's going all the way up, and it's going to innervate the spinal cord, okay? And that is a single cell. And these single cells, these individual neurons, can be bundled together into nerve cells. So there might be lots of sensory receptors in that big toe that all bundle together and send sensory information to that. But each one is a single neuron, not multiple cells. And so think about just an individual cell. If you are a tall individual, let's say someone who is 6 feet tall, I mean, that's a pretty huge cell, just to give you a little bit of perspective. And so in development, once we form that neural tube and we start to develop the nervous system, there needs to be some sort of information that is guiding where these things are supposed to go. You can see that up here in the higher parts of the spinal cord, these neurons that are exiting and entering the spinal cord are innervating or connected to the regions that are around that area. And so we don't have some sort of rogue neuron that goes from here that's going to go all the way down to the big toe, right? There would be a tangled mess if they all just went sort of willy-nilly. And so there must be some sort of a map, guidelines, directions during development that tell cells, these developing neurons, as they're exiting this developing spinal cord, where to go in the body. So there must be maps that tell these neurons down here to help go to the lower limbs, the ones up higher to go to the trunk, to the upper limbs, the arms, and then into the cervical region. It's a pretty amazing process, and that's what we're going to talk about. Let's take a look at that gastrulation to neurulation video that I showed one more time. So you can start to see the dorsal blastopore lip, and now things are involuting. You get that convergence and extension. You're going to get elongation. And now this is where neurulation starts. You're going to start to see these sides start to become columnar. They rise up. They will zipper up from posterior to anterior. The neural plate is formed. And now actually all sorts of things are happening. The spinal cord is formed. Got a little beam. And already there are nerves that are growing out towards the periphery and from the periphery back. One more time. So taking a look at this figure from the textbook, this is just giving you sort of an overview from two different perspectives of the process of neurulation. They're showing you what's called the early neurula, the mid-neurula, and the late neurula. And we saw this just in the video a second ago. And we're looking at it from both the dorsal surface in this top row and then a transverse section. And you can see where that section is here. It looks like the glass slide that's sort of cutting through. And then you're looking down through that there. So first, let's take a look at just some definitions. First, here is where the blastopore was, okay? And this region in here, this set of cells that are here, are what's called the neural plate, and we'll talk more about those in just a minute. The neural fold, I was trying to show you that in the video, and you might want to rewind to look at it. But the neural fold, you'll see these cells that are sort of rising up, and basically they're becoming columnar, like columns. Remember we talked about how cells change shape during these processes? So they're rising up, and eventually, excuse me, those neural folds are going to get so high, if you look over here on the far right, they join and meet, and then they fuse along the midline. And if you look at that in the lower set of images, you can see that here. So the neural plate cells are these sort of purpley, kind of grayish purple, right? And the ones that are going to become the neural folds are actually down here, okay, along the sides. I don't know if you can really see that color change there. But what happens is these cells, these light blue cells, again, become columnar, which make them taller, causing them to extend upwards. So now I'm in the middle figure here, right, like that. As they continue to form upwards, eventually, and the same thing's happening with these neural plate cells too. Eventually, what's happening, if you look at the far right, on the lower far right, those neural fold cells have now become the epidermis. They've folded over the entire top and connected. And that's shown if you look just above, that's where they zippered from top to bottom. And then enclosing then the neural tube, which is also sort of risen up. You can see from the middle here and formed a circular structure that runs the length of that embryo. And then just sort of coincidentally, we've got, here's our what will become the gut, the endoderm, and the mesoderm. Now we will talk a little bit more about the cells that are in here a lot more closely, okay, and what they do, who instructs, which cells are instructing other cells to do things, what are those signals, and so on. And so let's talk about those things now. This is a drawing I did when I teach neurobiology in the summer. This is some of the lecture notes of mine, and I thought I would just show it to you before we go into some of the details. It's very similar to the figure 13.4 in your book, which I'll show you next. Couple things about color coding here. The perspective epidermis, what will be the outer covering, or the skin, basically you could call it, of the developing embryo, I have here in this sort of green. Okay. What do you call it? The somites, which we'll talk about later, are these kind of gray stuff. That'll become bones and things and part of the spinal, not the spinal cord, but the vertebra. The neural tube, or the perspective neural tube, is the blue. Okay. And the red are going to become the neural crest cells. So one of the things, and what did I say? Oh, no, that's okay. Never mind. So let's take a look at this. So essentially what's happening here is these cells, the green, are pushing upward here and here. And they've got these several what they call hinge points. Okay, there's a midline hinge point here. So let me get a different color. My normal red. So you're going to have a hinge point as these are pushing up. The blue cells are sinking down. Okay. And there's a midline hinge point here. There's hinge points here and here. And so these are pushing upward like this. And eventually they're going to get to a point where the red come together, they're going to fall down. The blue are sinking, and then they will form a tube, the neural tube. And then the reds are going to fill in this area here while the epidermis is coating the entire region. This little circle down here is what's called the notochord, which is sort of defines that anterior-posterior axis. All right, so now let's take up the figure in your textbook. Now the book describes things, something called primary neurulation and secondary neurulation. For our purposes, I'm just going to talk about primary neurulation. I said I was going to pick and choose some things. And just for understanding processes, I think it's important to know one. So we're going to just focus on this part. And now this, once I get rid of all of that, you know what I should show is primary and secondary have to do with the different processes that occur along this neural tube formation. And here they have these terms called rostral and caudal. Rostral is going to be towards the head, or the anterior, caudal is towards the tail or the posterior. So we're focusing on things that are happening up near the rostral end or the anterior end. So getting rid of that. You'll see this is very similar to my hand-drawn image. We've got the epidermis rising up, which is they show in blue. We've got the sinking of the cells that will become the neural tube. That's sinking down. You get these two major hinge points here and here. They fuse together here. And then we get the epidermis extending over the entire surface. All right. So primary neurulation really has sort of four overlapping in both time and space stages. The first one shown here is called elongation. And so as you saw in the video of the Xenopus gastrulation neurulation, you saw that it was extending sort of like from, it started out sort of as a, almost a sphere. And then during neurulation, you would see it sort of extend along that anterior-posterior axis. Sometimes that's called the rostral to caudal, as I said before. And rostral to caudal, because chick embryos were primarily used to do a lot of this work, it was called beak to tail. Okay, it's the same thing, anterior-posterior, rostral caudal. So we're getting elongation in that anterior-posterior axis. Second, we're getting the folding. And I love the images that accompany these figures, these sort of cartoon figures. So these are chick embryos, electron micrographs. And so I mentioned to you about all these different cell types. And again, if I kind of come back here, remember the neural plate is going to be this sort of grayish purple. The epidermis, or what's going to be the surface cells, or the ectoderm, is the darker blue, and this sort of aqua blue is going to be the neural crest. So just keep that in mind here. Again, this is going to be the epidermis, neural crest. Here's our notochord. Again, the notochord is important for defining that AP axis. And this is where that mid-hinge point is. All right? And look at that. You can see this beautifully here. Here's our mid-hinge point. Whoop, let's not do that. Can't see anything. How about yellow? There we go. So here's our hinge point. And again, you can see it here, but there's going to be 2 more hinge points here and here that are not shown in the cartoon in terms of folding, but we'll get there. In the third stage, this is where I talked about the elevation of those neural folds, where they're sort of becoming columnar. And this is another beautiful image here where they're staining for different things that are inside the cells. And I can't remember what each color is for. And I can't even remember if they told you in there, but you can look it up in the caption of the figure. But you start to get this thing called the neural groove that's here. Okay. We still have our hinge point. But we're getting the elevation of the folds. They're coming up and rising up in this direction, like this and like this. And then lastly, well, actually, that's not quite lastly. We're going to get convergence. So we're getting this, all these rising up cells. And this and this, they're going to come together right here and here. They talk about the hinge points. Let me get a different color for this one. Here and here. They're called the dorsal lateral hinge points, or DLHP, dorsal, lateral. And it makes sense, because dorsal, we mean it's on the sort of the back of the organism, which is where this is happening, or the future dorsal side. Lateral meaning it's on the side. Hinge point. It's nice when something is called something that makes sense as opposed to the kind of crazy names for genes. All right. And then finally, we have here. So now what we've got is we've got that epidermal layer that is covering the surface. Here's our neural tube. It's closed completely. Okay. And then inside here, it's right about, well, let me get a different color again. So there are cells called neural crest cells that are right up in here, and they are going to start to migrate outward. And we'll talk more about them in a little bit. And at some point along here, it's also a little bit hollow. You can see on the cartoon here, there's a little hollow region. But that's basically what's happening, the 4 parts of neurulation. So now what I want to do is take a look at what are the signaling molecules that are involved in these sort of changes in cell shape and the movements of cells that induce neurulation. It's electric. She's a moving
bitch.